Good morning, afternoon, and evening, everyone. Thank you all for joining today's webinar titled The Impact of Maternal Obesity on Fetuses and Children's Neurodevelopment. My name is Maha Sheikh, and I am the Communication Specialist for the Jordan Nutrition Innovation Lab. As more attendees are joining the webinar, I will begin by going over some of the logistical items. Next, please. I would like to direct all attendees to a few functions on this Zoom call. At the bottom of your screen, you should see a chat icon and a Q&A icon. Please use the chat feature to engage in relevant conversation with the other attendees, and feel free to introduce yourself as well. Next, please. If you have a question for our speaker, please use the Q&A feature. We have allotted the final 15 minutes of this webinar for Q&A, at which point our speaker will respond to any questions from the audience. If you're experiencing any technical difficulties, please send a message to the chat specifically to host and panelists so that our team can assist you. This webinar is being recorded and it will be made available on our website following the event. We will repeat these technical housekeeping items in the chat throughout the webinar as people may be joining in at later times. Next, please. I would now like to introduce Haneen Abulayla, the Country Coordinator for the Jordan Nutrition Innovation Lab. We'll talk more about the lab and introduce our moderator for today's webinar. Haneen?
Yes, thank you, Maha. Good afternoon and good morning, everyone. My name is Haneen Abulayla, and I am the Country Coordinator for the Jordan Nutrition Innovation Lab. I am very delighted to open our second webinar for this year and happy to see more people joining our webinar for today. Before I introduce our moderator for today, I would like to give a brief introduction to the Jordan Nutrition Innovation Lab. Our project is a research and capacity building program that is funded by USAID Jordan under Feed the Future initiative. We pursue research and capacity building activities to support the health and nutrition of women of reproductive age, pregnant lactating women, infant and young children in Jordan. Some of our key objectives are implementing a rigorous maternal and child nutrition research agenda, including conducting analysis of existing nationally representative data sets and conducting comprehensive evaluations of programs such as USAID's Community Health and Nutrition Program. Moreover, we build individual and institutional capacity through awards of fellowships, scientific symposia, research workshops, and webinars. As part of the capacity building agenda, recently we have successfully implemented the second research methods and designs workshop in cooperation with Ministry of Health and USAID. In addition, we conduct these webinars on a regular basis as a means to conduct outreach to wider stakeholders group in and outside Jordan. This webinar is moderated by Dr. Lynne. I would like to introduce Dr. Lynne now. Dr. Lynne is Saqr bin Mohammed al-Qasimi Professor in International Nutrition, as well as a professor at the Friedman School of Nutrition Science and Policy at Tufts University. She has been an active investigator in USAID-funded projects in Malawi, Uganda, Nepal, and most recently in Jordan as part of the Jordan Nutrition Innovation Lab. She has been active in experimental studies with humans on lipoprotein, responses to several vegetable oils, trans fats, and soy protein, and the glycemic response as modified by various meal components. Most recently, she has been examining the molecular mechanism of action of carotenoids, both provitamin A and non-provitamin A, against several chronic diseases. Dr. Lynne, the floor is yours.
There we go. Good morning and nice to see everybody on. I have the pleasure of introducing Dr. Tomo Tarui. Dr. Tarui is a child neurologist and director of the Fetal neonatal neurology at Hasbro Children's Hospital in Rhode Island, the US. Dr. Tarui is an assistant professor of pediatrics and neurology at the Warren Alpert Medical School at Brown University. He is a physician scientist in developmental, neuroimaging, and genomics. His research focuses on developing non-invasive neuroimaging and genomic technologies to assess human fetal brain development and its clinical implications. Over to you, Tomo.
Thank you very much, Lynn, for the kind introduction, and to this whole research community and government community. I'm really honored to present my talk today and share our passion about fetal brain development and how it's relevant to maternal obesity. This talk is titled Impact of Maternal Obesity on Fetuses and Children's Neurodevelopment. And this is, I'm going to discuss anything, sorry, there is no financial relationship that I need to report. I won't discussing off-label uses for purposes other than that for which the FDA in the United States approves the product use. However, I will discuss the investigative analysis of fetal brain MRI beyond clinical practice. And this is the overview of today's talk. First section, I'm going to present impact of maternal obesity pregnancy on offspring children's neurodevelopment based upon the various reported studies. And second, I will overview how we can assess the living fetal brain without hurting them. And lastly, I would like to share our preliminary data on the altered fetal brain development on fetuses of pregnant women with obesity. And this talk, you may remember Dr. Perry Otirin-Jing, who gave the talk 2 weeks ago. She's my collaborator on this project, and there is a relevant connection between that. It's kind of like sequel of her talk, focused on neurodevelopmental impact in maternal obesity pregnancy. Needless to say to this audience, obesity pregnancy, maternal obesity determined by pre-pregnancy body mass index larger and equal than 30 kilograms per square meter is an epidemiological public health risk all over the world, including United States. About 29% of the pregnancy is complicated with maternal obesity. And various studies already shown maternal obesity has transgenerational health impacts on their offspring children's health, not limited to the neurodevelopment, but cardiometabolic health, etc. And this is the estimated distribution of obese pregnant women on the global perspective based upon the 2021 Strauss paper, largely based on Chen et al.'s data. As you can see, it is worldwide phenomenon. Offspring children of maternal obesity pregnancy have increased risk of cardiovascular, metabolic, neurodevelopmental, and psychiatric disorders. In this talk, we focus on neurodevelopmental impact of maternal obesity. A large-scale national registry studies reported that maternal obesity increases the risk of various neurodevelopmental disorders in offspring, such as attention deficit hyperactivity disorders, autism spectrum disorders, and anxiety and depression. And I'm going to talk a little bit more detail about these conditions. And ADHD, based upon the available literatures, the prevalence in Jordan is about 6.2 to 40.6%, depends upon the report. And the United States latest report is about 8 to 10% of children have ADHD. And ADHD manifests as hyperactivity, impulsiveness, and poor attention, thus resulting in impairment in working memory, executive function, and impulsive behaviors. It is pretty prevalent, and it's not a rare thing in a school system. As a parent, some of you may have experienced as well. the causes of ADHD, it's not really well determined. And people look at the genetic etiologies or environmental etiologies. There's no like single genes that strongly associated with ADHD. And it is still mystery. So it's so popular disease, why this ADHD occurs. And based upon the 4 Nordic birth cohort, which is a very large one, studies found that maternal body mass index dose dependently increases the risk of her children's ADHD symptoms. And especially excessive gestational weight gain increases the risk. So it's not only pre-pregnancy body mass index, but also how much weight, how aggressively the weight was being obtained during the pregnancy could increase the risk of ADHD. And this is a summary of those studies. In this table, left column is severity of maternal obesity, and right side is the percent increased risk of ADHD in the offspring children. In the group overweight, which determined BMI between 25 and 30, there's a plus 23 to 28% increase in offspring children. In obesity range between 30 and 35, 47 to 89%. And in severe obesity, which is BMI above the 35, there's 88 to 95% increase risk, basically double the risk of the ADHD in offspring children. And that has been shown not only in this Nordic birth cohort studies, but in other registry studies or cohort studies as well. Autism spectrum disorder, this is actually increasingly recognized in countries all over the world. In Jordan's data, I could not find the direct data, but it published data from UAE was in 2007 was 0.29%, and Oman 0.14% based upon 2011 study. However, non-published data I found in website, I couldn't cite here, shows much more higher incidence of the autism spectrum disorders in Jordan and those countries as well. In most updated data in United States, it's about 2.3% of the population, 1 in 45, which is pretty high risk, and which largely contributed by the diagnostic factors, means how much aware the physicians are, but also how much diagnostic studies are available, which is pretty not so much available because of very time-consuming and specialized testings for the ASD, including the United States. It is the bottleneck of the diagnosis of autism spectrum disorder. And autism spectrum disorder manifests as difficulties in communication, interaction with other people, restricted interest, and repeated behaviors. This is the big overview of the diagnosis. However, as the name autism spectrum said, it's just a pretty broad spectrum from mildly affected person to more severely affected person. Causes of the autism spectrum disorder has been vigorously studied in the recent decades, especially with whole exome sequencing. And there's a lot of genetic risk factors or genes even associated with the autism spectrum disorders. But there's also epidemiological studies mentioning that there are a lot of environmental factors affecting the development of the autism spectrum disorders, including the maternal obesity. The pre-pregnancy obesity increased the odds ratio for autism spectrum disorder in offspring by 1.3 to 2.05 fold, based upon the various studies looking at the association between maternal obesity and autism spectrum disorder in offspring children. And similar to ADHD case, excessive gestational weight gain also increased the risk for autism spectrum disorders. Depends upon the studies from 10 to 58%. So based upon those epidemiological studies, at least we could say maternal obesity may be a significant risk moderator of offspring children with neurodevelopmental disorders. More genetics, there can be a more genetic involvement in the disease development of those conditions. However, maternal obesity may give a more push to be symptomatic. And what is then impact of the maternal obesity on offspring's child's brain development? And some studies were already done in looking at the children and young adults of the offspring of the maternal obesity pregnancy. And they have structural and functional alterations in the brain based upon the MRI study. So in children and young adults of the maternal obesity pregnancy, they have smaller hippocampus in school-age boys, but not in girls. Altered white matter integrity, which I'm going to show later in the MRI studies, but it means higher fractional anisotropy and lower mean diffusivity, the measure of the MRI in children and adults. And because maternal obesity is the phenomenon during the pregnancy, there must be some impact in utero affecting the brain development. So people look at the younger generation, like neonates, the newborn's brain study, and it's very limited in the number. One study found poorer white matter maturation in newborns, newborns who were obese. There's also decreased functional connectivity in the dorsal anterior cingulate cortex based upon the connectivity study. However, information on fetal brain development, how the maternal obesity impacts on developing fetal brain, is lacking. To look at more mechanical aspects of this phenomenon using the animal models, such as mouse and rat models. They created rat models based upon diet-induced obesity during pregnancy. So they could control what to give to the pregnant mice or rat and look at the pups or fetuses brain development in the histology or the microbiological technology. And there, many studies were done, and it's found maternal obesity affects offspring's intrauterine brain development in mice and rats that is manifested in histology phenomenon, increased neuroprogenitor proliferation, altered neuronal differentiation maturation, and altered DNA methylation patterns on these pups. And how such histological changes occurs, people look at in detail, and it seems to be that maternal obesity make that changes through altered lipid metabolism, inflammation, and oxidative stress. So there seems to be biological evidence that maternal obesity altering the structure of the fetal brain and in relation to such molecular mechanism of altered lipid metabolism, inflammation, oxidative stress. In mice and rats models. And still, we have no information on human fetal brain development, how such changes, what kind of changes occurs, and how such kind of changes occur. In current stage of the medicine, we have certain ways to assess human fetal brain morphology and development. And that has been used in daily practice of the obstetric practice, especially when obstetric ultrasound that is routinely performed during the pregnancy detected some questionable fetal brain anomalies. Ultrasound is usually the first tools to detect. On the left side, there's anatomic diagnosis and genetic diagnosis on the human fetal brain development. And once the ultrasound, for example, in this case, it's the axial section of the fetal brain, showing significantly enlarged lateral ventricles, then usually such pregnant women referred to the MRI to have fetal brain MRI, and also the consultation to neurologists like me. On MRI, a pregnant woman is going to be wheeling from the feet, and their belly is going to be scanned, aiming to capture fetal brain. And in this case, the MRI found not only the enlarged ventricles suggested for hydrocephalus, but also fused thalamus, the thalamus, which is the subtle abnormality on the centerpiece of the brain development. Sometimes pregnant women undergo to the genetic diagnosis investigations, such as amniocentesis, colonoscopy sampling, and nowadays maternal blood. In this case, amniocentesis was done to collect the fluid surrounding the baby, including fetal cells, and did the genetic testing and found mutation in L1CAM gene, which is to be a stop codon mutation. So there is a functional dysregulation of this L1CAM gene. Made a diagnosis of congenital hydrocephalus syndrome or MATA syndrome related to this mutation. So this is how we assess human fetal brain morphology and genetic background in current clinical practice. By the way, the MRI is considered to be safe to both fetus and pregnant women. When we assess the fetal brain, it is very important to know that fetal brain is a dynamically developing tissue, organ. And it reflects their vulnerability, but also the potential as well. This is the scheme from the embryology website. Start with embryo at 8 weeks and then fetus throughout the pregnancy. The overall body gets bigger and change shape, but so is the brain. This is the 13th week of the fetal brain, computational imaging, and it increases in size, but also later in morphology, such as palpations occurs, especially after 29 weeks of the pregnancy. And not only that, this long journey of the brain development starts with the neuration, when the ectoderm's surface folded in to create neural tube, and top of the tube ballooned up to create future brain of the telencephalon. And at that time, the regions on the surface of the brain had started to be divided, related to the future functional regionalization. And also on this thin wall of the balloon, the neurodevelopmental cells, which is the source of the neurons, actively proliferate. And once they stop the proliferation, they migrate towards the surface of the brain to formulate cortex. And with those increasing numbers and with unknown mechanisms, especially in human fetal brain, started having complicated surface foldings called gyrification or sulcal development, and became at the term almost close to become adult brain. So this is a pretty dynamic process. And when we assess the fetal brain, assessing 25 weeks of brain and 33 weeks of the brain, they are supposed to look different, and we need to count in such differences as well. So in one word, fetal brain is a moving target process. I've introduced about fetal brain MRI technology that we use for clinical practice, but in research, people have been using more in-depth analysis, because this is, once it's acquired, this is a digital image. So using the digital imaging processing technologies, people have been analyzing fetal brain. I would like to introduce overview of such technology. So when fetal brain MRI is acquired, it has a lot of motion artifact, because it's all good things. Mother is breathing, mother is artery is pulsating, and fetus is also training, breathing and swallowing the amniotic fluid. It makes motion artifact, which is MRI is very sensitive. We use the post-acquisition computational processing to correct the artifact. Sorry. And once the motion is corrected, we align the fetal brain and then label the structures that we can recognize in the fetal brain, which is what we call segmentation. And once you do this labeling, you can stack together to calculate the volumes of each labeled structures. Also, using that data, we reconstruct the three-dimensional cerebral surface for the further analysis, such as curvature analysis or sulcal development analysis that I'm going to discuss soon. This all computational processing enables us to look into the fetal brain development quantitatively. So to assess, to determine the impact of maternal obesity on fetal brain development, especially in regional fetal brain growth, we conducted a prospective observational study at Tufts Medical Center. This was led by Dr. Emiko Takeoka, my lab's research fellow, and collaboration with Dr. Perry Otini-Jin, who was the last time speaker on this seminar series. And shared interest how maternal obesity influencing on mothers, also the fetus, and placenta. So inclusion criteria for this cohort is healthy pregnant women aged 15 to 45, singleton pregnancy, and gestational age at recruitment between 18 and 36 weeks, and both fetal sexes. We excluded if there's a multiple pregnancy, if there's abnormal fetal sonography findings, MRI findings we found, or non-chromosome abnormalities. So we look at the fetus and pregnant women as healthy as possible, besides the various range of the BMI. We offer the research fetal brain MRI scan because they are not really used routinely scanned for being healthy. And we conducted the segmentation volumetric analysis using the FreeView, which is the imaging analysis softwares. We conducted statistical analysis by comparison of the nonlinear growth regression models between BMI groups. We divided BMI groups between lean, overweight, and obese group. So again, this is the processing of the fetal images, as we discussed, with the low volume data motion correction, spatial alignment, and the segmentations and surface reconstruction. And this is actual brain image and structures, like we segmented cortical plate, subcortical structures, ventricles, brainstem, and cerebellum. And this is a result. This figures has all designed as the x-axis being gestational age, y-axis in volume of that particular regions. And this is the nonlinear regression models. And we found left cortical plate, right cortical plate, both cortical plate, and cerebellum. These square dots, overweight obese group fetus have accelerated growth on these structures compared to the controls, especially after 28 weeks of the pregnancy. We look at the, is it like specific that cortical plate is getting bigger, or is just merely reflection the whole brain is getting bigger? So therefore, we calculated the ratio between cortical plate and the whole brain and plotted in gestational age in the x-axis, and found that indeed, actually, after 25, 28 weeks, cortical plate had specifically accelerated growth in comparison to the whole brain. The cerebellum, we didn't see clear patterns. But that tells us that the cortical plate is really, maternal high BMI has increased regional brain volume, especially cortical plate and whole cerebellum. And especially cortical plate has increased volume compared to the rest of the brain. So there's a specific change in their structures in cortex. The current study still has limitation because it's a small numbers. We need larger cohort study, and we need to have a longitudinal analysis. In this case, we only plotted the whenever the pregnant woman agreed to do the MRI one by one. But ideally, we should follow up the same subject, 2 MRI, 3 MRI, so we can have the trajectory. And more importantly, we should look at associations with neurodevelopmental outcomes. Because the bigger brain is not necessarily beneficial to the person. We knew that from infants, toddlers with autism spectrum disorder. They have a usually larger brain. And they later on, the larger brain actually became less prominent as they become older and even become smaller in general after adulthood. And children and adults with autism spectrum disorders have altered connective gyrification. So remember that the not only the growth, but afterwards, like wiring of the brain is and gyrification, such later phase of the brain development is also altered. So we need to have more comprehensive view on the brain development, how it was impacted by the maternal obesity. And that study is currently undergoing. And how comprehensively we can look at tissue of the brain. I mentioned about connectivity, diffusivity, which I'm going to explain now. We can use fetal brain diffusion MRI to assess axonal connective and connection development. And diffusion analysis, we can get the measures called apparent diffusion coefficient, fractional analysis. These data we get in the clinical scan as well. But also for research scan, we can get more advanced diffusion imaging, such as diffusion tensor imaging. The diffusion MRI is looking at the water molecules' diffusivity or mobility. In like the cup of the glass, the water molecules, for example, is moving freely in all directions, X, Y, Z axis. However, water is packed inside like the fiber bundles, such as like white matter axon bundles. The water molecules' mobility is more free on the longitudinal path, but limited to the tangential path because of the packed fibers. That causes anisotropic diffusion along the fiber tracks. And diffusion MRI detects such anisotropies. And for example, this is a raw MRI diffusion signals. And this ellipsis represents the anisotropic diffusion of each dot. And using the computational refinement, it can eventually reconstruct the fibers indirectly using the water molecules diffusivity. So using this high resolution diffusion tensor imaging, you can actually reconstruct the fiber architecture in the brain. And that was done in fetal brain, so it's very challenging because the diffusion images is sensitive to the motion artifact. But Dr. Kasparian's group in Vienna did beautiful studies to look at the diffusion-based fiber tractography in living fetus, and also that same fetus, when they get the pathology specimen, they look at the pathology. They also did the MRI diffusion tractography of the pathology specimen. And left side is the typically developing fetus. And on the living fetus, you can see the fiber reconstruction presenting the vertical fibers running in the white matter, subcortical white matter, also the transverse fibers of the corpus callosum. And this is the similar things in higher resolution is seen in the pathology specimen of the MRI. You can see the fiber tract like corpus callosum and also the subcortical white matter fibers. And it's consistent with the histology findings that stain the fibers connection. In right side, this is the fetus with agenesis corpus callosum, the missing the corpus callosum connecting to halves of the brain like here. And they also see such abnormal fiber alignment seen in the agenesis corpus callosum in the MRI. This is the living MRI and diffusion reconstruction. You can see the fibers are here on two halves, but not connected clearly between. And in the pathology specimen, it's beautifully reconstructing the fiber structures, but it's not crossing here. And same for here. It can actually see the probus bundle, which is the abnormally running fibers, which couldn't cross in these MRI and histology consistent. By the way, you can see these radial column signals in the cortex, where there may be fibers, but this actually more represents the calamic cellular alignment. So important thing to know is the diffusion signals doesn't specifically show the fibers, but it just shows aligned structures, whatever it is, either fibers or cell column. My collaboration with Dr. Kihoin, we developed the technology to assess the Saqr pattern development of the surface of the fetal brain. Dr. Kihoin is investigator in Boston Children's Hospital. And his labs developed the technology to extract the surface of brain, determine this pores represent the sulcus pit, the deepest part of the sulcus, representing the position, and also measuring the depth of the sulcus and area of the circle basing. He can quantify the various features of the sulcus and compare the sulcal position, for example, between the corresponding sulcal basins, or compare the sulcal position intersulcal relationships, which is important because we align the brain, they may actually obscure the sulcal position, but intersulcal position is not disturbed, whatever our alignment is, because it's between two sulci. And what we eventually get is the calculated similarity index. Similarity index is the compared our subject brain, either case or control. We have the database of the normal brain template. We compare how similar they are to the normal archived brain. The value ranges from 0 to 1, 1 be 100% match, and 0 is totally no match. Usually, normal value represents distribute between 0.82 to 0.9. And we use that technology not for the maternal obesity yet, but the condition called isolated agenesis corpus callosum that I briefly mentioned. And this is the reconstructed surface of the fetal brain. And this is the normal control, and this is the fetus with agenesis corpus callosum missing the connection between two halves of the brain. And that should be the only features of the abnormality. However, we found actually the surface feature, sulcal development pattern seems to be quite different. We can see with naked eye a little bit, but we can't really specifically say what it is. But based upon the sulcal pattern similarity analysis, which I showed examples here, the distribution of the agenesis corpus callosum group, it's usually much lower compared to the normal groups throughout the gestation. There's also the surface analytic method called gyrification index that has been historically used. We didn't see any difference in that measures. So because we think we are really looking specifically in sulcal position, not like curved features of the surface, that's the reason we could detect such differences. This is we can also look at the region, particular part of the brain, sulcal developmental pattern, especially like sulcal depth. Conducted the analysis of the fetus with Down syndrome. We did a fetal MRI for the fetus with Down syndrome and analyzed the surface features. In this study, this heat map shows, in comparison to typically developing fetus with Down syndrome has different sulcal depth significantly in this red accentuated region, which is the left bilateral sylvian fissures, left superior temporal sulci, here, this one, and right central sulcus, and right palliate occipital sulcus, just here. And why it's important? It's because the surface of the cerebrum and cerebral surface has the regional differences in their anatomy, but also in their future function. Like people know, there is a language area in the left frontal lobes. There is a visual cord field at the back of the occipital lobe. So regions of the cerebrum have different function in the futures. And when we look at these regions and related functionality, such as executive function, multimodal processing, auditory language and memory, fine motor function, and visual motor processing, these red areas have those functions. And actually, it's pretty consistent with the developmental phenotype specific to children with Down syndrome. In simpler words, the children with Down syndrome has weakness on these areas compared to the typical developing children. So therefore, it may be possible that in the fetal brain, we can already see the difference in the future functional variation. In summary, maternal obesity increases risk of offspring ADHD or autism spectrum disorder by 1.5 to 2-fold. Human brain imaging studies, animal model studies suggest interesting origin of fetal brain development. And quantitative analysis of fetal brain MRI revealed maternal obesity increased region-specific fetal brain growth. A multimodal fetal brain MRI analysis can assess comprehensively human fetal brain development. And that's what we are trying to do as a next step. Because understanding impact of maternal obesity on fetuses, children's neurodevelopment may open doors for prevention and intervention. And it's not really so obscure idea because people have been looking at the way to improve neurodevelopmental disorder by nutritional intervention. For example, the fatty acid has been trialed in many studies as a treatment of neurodevelopmental disorders. Because, for example, long-chain polyunsaturated fatty acids have been thought to play critical nutritional roles in various neurodevelopmental and psychiatric disorders, especially in ADHD. In just observatory studies in children and adults with ADHD found lower N3-LCTUFA, basically omega-3 fatty acid in blood levels. This is not related to any maternal obesity pregnancy. And 9 randomized controlled trials in 2012 tested efficacy of these omega-3 fatty acids on ADHD symptoms. The supplement kits with that. And 7 out of 9 studies reported some improvement in ADHD symptoms. And if you remember 2 weeks ago, Dr. Thierry Jean's talk, this omega-3 fatty acid is a key players or key changes occurred in pregnant women and placenta of the maternal obesity pregnancy. So these are unrelated studies, but we start connecting dots. This must be something that lipid metabolism may contribute not only metabolic changes in maternal obesity pregnancy, but potentially influencing on fetal brain development and associated neurodevelopmental disorders. I would like to introduce and appreciate to my collaborators, and especially children's family participated to the study. This work is largely done at Tufts Medical Center, where I used to work until November last year. And my collaborator is still there. I have a lot of roles onto that. Thank you.
Thank you very much, Dr. Tomo. That was fascinating. I think now is the time for questions, and I think we've already had a couple of them coming in. First of all, Amanda Pierce wonders, what is considered excessive gestational weight gain?
Yes, well, it's more like the obstetricians, researchers' expertise than mine, but there is expected weight gain, depends upon the countries and their criterion. And usually researchers look at if it's like within recommended range or above that recommended range. And it's also percentage increase in maternal weight. Of course, you should, pregnant women should increase the weight throughout the pregnancy because amniotic fluid and fetal growth, everything. But if it's beyond that expected range or not, means there is also increased risk of the maternal obesity acquisition during the pregnancy. That's how people assess and quantify for the research.
All right, thank you. Haji Aman said, is there an alternative way to assess this effect in resource-limited areas where they don't have MRI and other advanced equipment?
Yes, the ultrasound is actually might be more available than the MRI. And nowadays, ultrasound becomes really, really high resolution. So using that might be one way. I'm actually very much interested in doing that to get the better ultrasound image quantified. But also assessing the newborns fetal growth in general, but also the newborns adiposity, newborns metabolic status may also, and then follow up the child's neurodevelopment. Such like the way we could also, such data is still missing as well to precisely describe how maternal obesity affects the longitudinal child development. So developmental testing, it will be a very important thing to do.
All right, Judy Kanhwadi says, since overweight and obesity increases the risk, but doesn't affect the neurodevelopment of all fetuses, is there some insight into what is protective, aside from long-chain fatty acids?
That's a very interesting question, actually. And certainly to look at that, well, there's of course the various factors like maternal nutrition in general. That's more your expertise, I think, Lynne and everyone in here. What to eat is a different, also the important, not just the omega-3 fatty acid, but what other diet influence on maternal obesity would be also important. In terms of protective effect, I don't know if Dr. Thierry presented that in last talk, like how you get omega-3 fatty acid might be also important, like in comparison between, in her studies, comparing the Hawaiian pregnant women and Ohioan pregnant women. And there, of course, the baseline omega-3 fatty acid level is quite different. But how you get that through the fish or through the pill may also impact as well. Sorry, I don't think I directly could specifically answer your question, what can be protective factors. But to understand that, we need to know what exactly people are eating without obesity, because obesity, the BMIs marry the weight measurements. And people know this is not sufficient to assess the nutritional status of pregnant women in more comprehensively. So.
Okay, very complicated. All right. Another question is, what is more serious on brain development according to research studies, a pre-gestational obesity or that of excessive increase of gestational weight gain?
I don't have clear answer to that. And my cohort is certainly so small that I cannot really distinguish them. And but people may have answer, but I'm not aware of that.
All right. Thank you. Amani Hamdan said, is the risk only occurring in the first pregnancy mostly? And if so, does it increase in the following pregnancies?
It is each pregnancy kind of like the renewal at risk. Of course, I didn't do that study, but my colleagues and my former colleagues, Dr. Catalano, is looking for like the pre-pregnancy weight and inter-pregnancy weight gain as the potential risk factors of the metabolic changes in pregnant women. So, it's not only the first pregnancy, but as the second pregnancy, third pregnancy, it must be the unique impact onto the maternal metabolic health and also child's developmental health. However, I'm not aware of the studies that is really looking at that because it takes really long time effort study.
All right. Maj Afghani asks, could maternal obesity be hindering ketone body production that is essential for fetal brain development? Is there a therapeutic role for exogenous ketones in pregnancy?
I don't know the study that's particularly looking at the ketones, and so not knowledgeable. I know the ketone is the important fuel for the brain, but how it plays a role during pregnancy. Because a lot of study was done, for example, omega-3 fatty acids, how important to the fetal brain development, but I'm not aware of about the ketone. Yeah. But important things to look at because it's important, how to say, style of the lipids to get in energy source get into the brain.
All right. Noel Henrich asks, do you have any thoughts as to why the hippocampus is smaller in boys versus girls?
Well, the sex effect is everywhere in terms of the brain development. And there's also the sex differences in terms of the susceptibility to maternal obesity as well. For example, in my studies, male fetuses sometimes more affected compared to the females. And like the high-fat diet obesity dams in the mouse study, rat study, they did a neurodevelopmental testing of the pups of the affected groups, and females sometimes do worse. So I don't know the mechanism, but it is actually consistent theme that there is a sex differences in terms of the effect of the maternal obesity on the brain development. Well, at least in animal models.
All right, thank you. Abdul Halik Wakicho asks, was the pre-pregnancy maternal weight considered while you look at the effect of gestational weight gain?
Yes, the people has been looking at the pre-pregnancy BMI be very, very important. not only controlling the weight during pregnancy, but actually before get pregnant, it's important because it's already changed the metabolism, actually already primed the metabolism to be different. So yes, answer is yes, it's important, and I expect predict that it's same for the impact on neurodevelopment as well. I'm a pediatrician. In my opinion, this maternal obesity pregnancy should be first intervened in high school before the girls or boys get pregnant or make pregnant, because they need to be aware of how their health impacts the fetus. Because sometimes it's hard to change the behavior of the adult.
That's true. Okay, Haji Aman says, can you tell us how maternal undernutrition affects brain development?
I cannot tell, but I do know it's bad. And then because I didn't study, but for example, my home country, Japan, is facing concern of the maternal undernutrition. So my work is not really helpful to Japan at all, but they are studying that effect. And also in historical studies, in terms of based upon the natural history studies, based on significant food deprivation during war and so on, the metabolic changes occurred in offspring and children has been reported. I don't know in detail about neurodevelopment, but I would predict that will be also affected as well.
All right. Well, I have a question. You mentioned altered lipid metabolism, inflammation, and oxidative stress in the mother. What are the markers of inflammation and oxidative stress in women that you measure? And can you potentially find these in human brain of a fetus? Have any animal models studied this?
Yes.
I mean, do you look at CRP or cytokines, or what do you look at?
Yes. Well, first, I didn't do the studies. It's really the citation for the animal studies that people are doing. And yes, it is cytokines that people have been looking at, interleukins and so on, in maternal blood, and also the DAMS tissue as well, in the animal models. And inflammation also, I do think there is no evidence, but I do believe there is a role, because the children with autism spectrum disorders or animal models of the autism spectrum disorders, neuroinflammation seems to play an important role. The story is going to be a little bit not related to this topic, but like person's intestinal bacterial flora and its relationship with the children with autism's behavioral spectrum has been started reported. So people microbiome change, and also how like the interleukins and such inflammatory molecules changes in the brain, and also in this maternal obesity pregnancy, they're looking for placental inflammatory markers, how it relates to the child.
Are there any way that you can use labeled, I was going to say labeled products, stable isotopes, to see biochemical markers in brain?
Yes, in neuroimaging study in adults, especially for cancers or like tuberous sclerosis, certain like condition that they need to find the lesion to be active for the epilepsy surgery, etc. People use that for fetus. I don't think we legitimate to use the isotope tracers, so we may not be able to use for the fetus. But people have been using that in adult studies if the safety is well established for the new imaging studies, so looking for where the inflammation is, where the particular molecules are, for example, tuber-related molecules are.
Okay. All right. Sarah Salman asks, does this research mean we have to decrease maternal weight pre-pregnancy in a case of obesity? I mean, this is the big question, right?
it's a big question, and to answer that question, there needs to be layers and dimension of the research to do that. But in general, the current practice is to prevent excessive weight gain during the pregnancy, but not the undernutritioned pregnant woman, have more like the appropriate weight gain. That will benefit the overall health of the pregnant woman and also her offspring children. That is all I can say at this moment. To say that this is really important to control the maternal weight, to have the better outcome on the neurodevelopment offspring children, we need to do much more studies and larger scale human studies.
All right, Sigal Biyazan asks, why do you exclude multiple pregnancies from your study?
Because the twin pregnancy already has unique brain development compared to singleton. So that can actually certainly doubt interpretation.
All right. Another question, could you specifically say what the biomarkers are to identify oxidative stress in the women?
I do not know the biomarkers for the pregnant women. For example, you can detect on the maternal serum. There must be, but I'm sorry, I'm not aware. What I found in the literature is more for the like tissue level of the study, like placenta or of the human or animal studies. They look at like the gene expression of the oxidative stress molecules. That's the way they actually detected there is more oxidative stress or not on the maternal obesity. But I'm not aware of like biomarkers that you can check in the maternal blood.
Okay. Hanan Al-Jaifi says, does maternal obesity affect the epigenetic regulation of genes involved in brain development and function in fetuses and children?
That I would love to do the study for with this moment, specifically looking for neurodevelopmental genes methylation patterns. The people have been looking at the overall methylation patterns, and they found the difference, I believe, in metabolism-related gene methylation patterns. But I would like to explore that more for the neurodevelopmental genes and neuroinflammatory genes.
Are there any other questions? All right, I think then this has been a fantastic seminar, and certainly we all have a lot to learn, and many of us will go back to the recording that will be online in order to look at some of the slides again. Clearly, you've shown that obesity is associated with many adverse outcomes. Down syndrome, ADHD, autism, changes in brain size, probably. You didn't mention anything about correlations with cognitive development in children, maybe age 6 months to a year. I don't know how you would test brain function if you had scans of these fetuses before they were born, obviously. Is there any correlation between a scan and then the outcome in a really small child? I guess that has to be the last question.
Yes, you mean the very small child with the maternal obesity, or in general?
Yes, with maternal obesity.
Yes. Well, that part is exactly the missing information. How such first of all, there's no. We are among the first to do the studies to really look at the brain structural changes in fetus. First, we need that, and then we are currently following those children to see if there is any correlation. That is the answer to your last question about how this brain and development outcome correlate. And the impact of the maternal obesity on the cognitive function has been reported. Sorry, I didn't mention that. There is a slight drops in the IQs and so on, based upon the developmental testing. By the way, people have been trying to see if supplementation in general and for any pregnancy helps of the omega-3 fatty acid supplementation helps to child's various health outcomes, including cognitive functions such as IQ. For general obstetric population, there's actually Cochrane reviews went over many studies and so on. They didn't find significant benefit of the omega-3 fatty acid supplementation to increase the cognitive like IQs of the offspring children. Important thing though is this is for general population. For all pregnant, they supplement and did a trial. I couldn't really find the omega-3 fatty acid supplementation specific to maternal obesity pregnancy, and that could change based upon the available data that because significant correlation between the low omega-3 fatty acid and maternal obesity pregnancy, and low omega-3 fatty acid between and children with ADHD or autism. So it is possible that the IQ or other markers of the neurodevelopment may improve with the supplementation if we choose the right supplementation and if we choose the right population like maternal obesity pregnancy.
All right, thank you very much. I think those of us listening here have our work cut out for us to figure out how to implement something to decrease the rate of pre-pregnancy and gestational weight gain. And other than that, I'll say thank you very much for this, wonderful talk. And again, this will be posted online for those of you who want to hear this again. And if people have specific questions, I'd be happy to receive them and see if I can find an answer from somebody. Okay, thank you.
Thank you very much.
Yeah, thank you.